Swayam Prabha. Digital India. Educated India.
Today we are going to discuss about signal transduction pathways. Now, why do you have to talk about signal transduction pathways? Now, so far we have discussed how messenger RNA synthesis is regulated inside the nucleus. We took about 12 lectures to discuss various aspects of regulation of messenger RNA synthesis by RNA polymerase II. Then we spent some time trying to understand how the ribosomal RNA synthesis is regulated by RNA polymerase I, and synthesis is regulated by RNA polymerase I. Then we discussed about the synthesis of transfer RNA and 5S rRNA by RNA polymerase III inside the nucleus. So, all this time we have confined ourselves to only the nucleus, that is how gene expression is regulated inside the nucleus, how the chromatin environment influences the nucleus, epigenetic regulation of gene expression, and so on and so forth. But what we have to now do is to come out of the nucleus and then see how events which are taking place outside the nucleus influence the gene expression inside the nucleus, because cells are not alone. Cells are always either communicate with each other or they communicate with the environment. So, all the gene expression that we have studied so far happening inside the nucleus, it has to be orchestrated by events taking place outside the nucleus. So, unless we understand how does these genes or how does the chromatin and the genes inside the nucleus communicate with the external environment, and how stimuli either arising extracellular space or extranuclear space, they influence gene expression, and that is what the crux of the entire regulation of eukaryotic gene expression. So, how signals emanating in the environment as well as signal emanating outside the nucleus, that is in the cytoplasm, influence gene expression and regulate gene expression. This is what is the entire area of gene expression is all about. So, after understanding the basic mechanistic aspects of eukaryotic gene regulation, especially the three major forms of RNA, namely transfer RNA, ribosomal RNA, messenger RNA, now we will come out of the nucleus and ask the question, how signals emanating outside the nucleus and in the cytoplasm influence gene expression changes. So, regulation of gene expression by signals emanating outside the nucleus is what is going to be the focus of the talk, maybe in the next about 5 to 10 lectures. Now, when we talk about gene expression, there are basically two types of gene expression changes. One is constitutive gene expression and inducible gene expression. So, the transcription of genes inside the nucleus can lead to the synthesis of molecules that perform housekeeping functions. When I say housekeeping functions, it means the basic cellular processes that take place in all different kinds of cells. So, for example, if you take a higher eukaryotic organism, whether it is a liver cell or a brain cell or a muscle cell, all the cells now have to need energy for their metabolism and survival. So, basic energy metabolism pathways, that is the enzymes involved in basic energy generating pathways and basic metabolic pathways like glycolysis, Krebs cycle, electron transport, oxidative phosphorylation, these things have to happen in all the cells irrespective of whether it is a brain cell or a liver cell or a muscle cell. So, the genes which are making these proteins or making the molecules involved in this protein are turned on all the time irrespective of all the cell type is. So, such kind of genes which are required for basic aspects of basic survival of the cell or basic functioning of all the cell types are known as housekeeping genes. So, such genes are supposed to be constitutively expressed and they are turned on all the time in the cell, although their levels may be modulated in response to various cell stimuli. Whereas, genes whose expression is cell type specific or tissue specific, or genes which are turned on only in response to certain stimuli emanating outside the environment or outside the nucleus, they are called as inducible genes. So, we have two types of genes, one is constitutively expressed genes and inducible genes, and all the regulation takes place with these two kind of categories. Let us give one simple example before we go to high complex eukaryotes. Let us take a simple example from the simple eukaryote like yeast and then ask the question, how does the yeast cells respond to a change in the carbon source in the medium. As you know, we normally grow yeast cells like Saccharomyces cerevisiae in glucose as a carbon source, but suppose if we change the carbon source from glucose to let us say galactose, what kind of gene expression changes take place. Now, So, what we will discuss maybe in a couple of minutes is how does transcription regulation takes place in response to galactose. If glucose is changed to galactose in the medium, how do the yeast cells respond to this kind of a change in the carbon source. Now, there are about half a dozen genes which are involved in galactose metabolism, and many of these genes are either present on the same chromosome or they are present on the different chromosome. For example, the 3 genes I have listed here, GAL7 is present on chromosome 3, GAL10 is also present on chromosome 3, whereas on GAL1 which is also present in chromosome 3. On the other hand, genes like MEL1 which is involved in melobiose metabolism is present on chromosome 3, whereas the GAL2 gene is present on chromosome 12 and the GAL3 gene is present on chromosome 4. So, what I am trying to say here is that there are about 6 genes involved in the galactose metabolism, and these genes are located on different chromosomes. So, if the galactose metabolism has to be turned on in response to the presence of galactose in the medium, then these genes located on different chromosomes have to be coordinately switched on, and this is achieved by 2 transcription factors called GAL4 and GAL80. Now, the GAL4 serves as the positive regulator of gene expression. I think in the previous lectures when we started discussing about transcription factors, DNA binding domains and transcription activation domains, we have discussed extensively the GAL4 protein of yeast, which has been extensively studied and one of the best eukaryotic transcription factors which have been studied well. The GAL4 protein acts as a positive regulator of galactose metabolism, and the GAL80 protein shown in red here acts as a negative regulator of galactose metabolism. Now, let us see how these genes are turned on. All these genes, despite being localized on different chromosomes, in their promoters contain an enhancer element known as GAL4 upstream activation sequence, shown as yellow here. So, this upstream activation sequence is specific for all these genes involved in galactose metabolism, although there could be some minor variations. Now, what happens? As long as you grow yeast cells in the absence of galactose, the GAL4 and GAL80 interact with each other and they bind to the promoter element, especially the GAL4 UAS, and inhibit the transcription of the galactose metabolism genes, which makes perfect sense because there is no need to turn on the genes involved in galactose metabolism when there is no galactose in the medium. The moment you add galactose to the medium, or the moment the glucose is replaced by galactose as carbon source, the GAL3 gene metabolizes the glucose, and this metabolite of galactose now acts as an inducer, and this inducer now prevents the binding of GAL80 to the interaction of GAL80 with GAL4, and as a result now GAL4 acts as a positive regulator and turns on all the genes involved in galactose metabolism. So, you can see by simple change in the carbon source from glucose to metabolism, the gene expression inside the nucleus is turned on or turned off, and glucose is there, the GAL4 and GAL80 interact with each other, and all the genes involved in galactose metabolism are not expressed. When you change the carbon source to galactose, because cells now have to metabolize galactose, this signal is passed on to the nucleus, and all the genes involved in galactose metabolism is turned on by inhibiting the interaction between GAL80 and GAL4, and GAL4 now acts as positive regulator and turns on all the expression of the genes. So, this is one simple example just to illustrate. Although we have studied so far all the gene expression, organization of genes and the expression of the genes at the nucleus, all these things have to happen in coordination with the changes that is taking place outside the nucleus, and one simple example is how, for example, yeast cells respond to changes in the carbon source. Now, the enzymes involved in galactose metabolism and transport in yeast cells are inducible and co-regulated, even though the genes are localized on different chromosomes. So, this is unlike what we have seen in E. coli, where all the genes involved in a particular metabolic pathway are often present together and organized in the form of operons. Now, in higher eukaryotes and even in simple eukaryotes, this kind of operons seldom exist. But despite being localized on different chromosomes, the genes of a particular metabolic pathway are coordinately regulated because they all have common cis acting elements to which a common transacting reactor binds and activates the expression of a number of genes. All these genes involved in particular metabolic pathway. So, although the eukaryotic nuclear genes are not arranged into operons, they are coordinately regulated in the cell, and I have given you the galactose metabolism as one example. There are umpteen number of examples as we will discuss more and more about it. Now, let us come to complex eukaryotes. Now, the simple eukaryotes like yeast are single cells. So, most of the time the major communication they have to do is with the extracellular environment. But around 1 billion years ago, the ability of cells to communicate with extracellular signals took a great leap in complexity when eukaryotic cells began to associate together and started evolving as a multicellular organisms. So, it became clear because when these cells started coming together and then started organizing in the form of tissues, various cell types, it became necessary for them not only to interact with the environment, but they also have to interact with each other. So, cell-cell communication as well as communication of cells with the external environment became very, very important. So, along with this evolution of multicellularity came cell specialization, whereas in the case of simple eukaryotes like yeast, we only have a single cell type, but as the eukaryotes became multicellular, each cell became specialized. There are cells which are specialized for carrying out a cardiac function, cells which were specialized for carrying out endocrine function, cells became specialized to become a nervous system, and so on and so forth. So, as the cell specialization as well as development of various tissues organisms became evident, it became much more necessary to develop complex communication systems and complex regulatory systems to coordinate all these activities together, so that the organism can function in a coordinate manner. So, coordination of the development and environmental responses in these complex multicellular organisms required an array of signaling mechanisms. So, the signal transduction pathway, the signaling mechanisms, that is the signals which regulate gene expression inside nucleus, became more and more complicated. At least two important communication systems were developed in the multicellular eukaryotes. One is the nervous system, another is the endocrine system. So, the gene expression that is taking place inside nucleus have now to respond to the changes taking place outside the cell through these major communication networks such as the nervous system and the endocrine system. So, it became necessary to regulate gene expression inside the nucleus of a multicellular organisms in response to a variety of signals that are generated either within the cell or those that came outside the cell. This is what we have been telling now. Exerted their effects on gene expression by means of a series of biochemical reactions called signal transduction pathways. These signal transduction pathways greatly amplified the original signal and ultimately resulted in the activation or repression of genes inside the nucleus. So, this is what the need for a complex signal transduction pathway is a necessity, because as the simple eukaryotes like yeast evolved into multicellular complex organisms, in order to respond to these external cues, external signal stimuli, cells developed complex signal transduction pathways, so that the signal can be properly amplified and channeled through a specific pathway, ultimately leading to activation or repression of specific genes. And these signal transduction pathways often make use of proteins known as receptors, which act as an entry point, and it is the receptors which actually interact with specific signaling molecules, and these receptors are either present on the plasma membrane or they are present inside the cells. And the entire signal transduction pathway is governed by a series of phosphorylation and dephosphorylation events, which are basically governed by protein kinases and protein phosphatases. So, what we are going to now begin to understand is that as organisms became multicellular, it became necessary to evolve complex signal transduction pathways, so that gene expression can be either activated or repressed in response to specific environmental signals. And these signals can either act at the level of cell membrane, or they can diffuse into the cytoplasm and interact with these signaling molecules, interact with specific receptor molecules, and then ultimately through a cascade of phosphorylation dephosphorylation events, ultimately this entire signal transduction pathway culminates in the activation or repression of transcription of specific genes. Now, the enzymes which catalyze the transfer of a gamma phosphate from ATP to specific protein molecules are known as protein kinases. By this time, I am sure all of you are aware of what protein kinases are, because we studied extensively how phosphorylation of histones regulate gene expression and so on and so forth in the previous lectures. These protein kinases play a very, very important role in the signal transduction pathway as well. So far in earlier lectures, we studied how phosphorylation of histones can regulate gene expression inside the nucleus. Similarly, the protein kinases even outside the nucleus, by specifically phosphorylating and specific receptor molecules as well as other proteins, can ultimately influence gene expression. The protein kinases represent one of the largest families, which may consist of up to 2000 members in a eukaryotic genome, and even as I speak today, we still do not understand the exact physiological significance as well as mechanism of action of many protein kinases inside the eukaryotic cell. So, one of the major challenges of the 21st century is to understand how these various signal transduction pathways coordinate with each other, they work with each other, interact with each other, ultimately leading to activation of a specific gene expression program. This has been one of the major challenges, and we still have not achieved success in many of these signal transduction pathways. So, more than 80% of the cellular events are regulated by protein phosphorylation. And the three major residues in a protein which can be modified are serine, threonine and tyrosine, and these are the three residues which are targeted by the protein kinases, which we have already studied in the case of the histone gene regulation. But the regulation of gene expression by phosphorylation by histones primarily involves serine and threonine residues, whereas when you come to signal transduction pathways, the tyrosine plays a very important role, and in fact there are a group of kinases known as tyrosine receptor kinases which play a very important role in a number of signaling pathways. Phosphotyrosine plays an important role in molecular recognition and formation of a protein-protein complex, and phosphorylation at serine or threonine induce often conformational changes on target enzymes due to change in the repulsion. The charge-charge interaction lead to repulsion and result in conformational change, and this triggers a cascade of events ultimately culminating in activation or repression of specific target genes. So, signal transduction leading to the regulation of gene expression is characterized by a maze of complex intermolecular and intramolecular interactions. I am trying to prime you to the complexities that we are going to face at the later stages of this lecture series, because as I said, these signal transduction mechanisms have now become very complicated, and as I speak we still do not understand the exact mechanisms that are involved on the interplay of the various factors ultimately leading to the activation of a specific gene expression program. So, not only each signal transduction pathway operates and activates a gene, often a single gene, the activation or repression of a single gene depends on multiple signals and multiple signal transduction pathways. So, a lot of research is now going on to understand how the signaling molecules or how the signaling pathways communicate with each other, and as a result of this complex intermolecular communication as well as intramolecular communications, how specific genes are either activated or repressed. What we will now study and focus primarily is, at least in the initial part of this lecture series, is to understand the role of the endocrine system and the role of the hormones. How hormones, which are very important chemical messengers that have evolved in complex multicellular eukaryote molecules, how these hormones ultimately activate or repress the expression of target genes. As we all know, hormones are primarily produced by the endocrine system of the multicellular eukaryotes, and these hormones became very important players in the regulation of gene expression through specific signal transduction cascades. Now, so when we talk about hormones, which are chemical messengers which are secreted by specific endocrine organs in the body, and they then move to another place and then exert their effect. This is what the simple definition of a hormone, which most of you have studied at a very early stage of your education. So, these hormones which are secreted by our endocrine system of the body, there are primarily of two categories. One are called the lipophilic hormones, and these lipophilic hormones are capable of moving. Because of their hydrophobic nature, they can simply diffuse across the cell membrane or the plasma membrane and enter the cell, and inside the cell or inside the cytoplasm or in the nucleus, they can interact with specific receptor molecules, ultimately lead to the activation or repression of specific target genes. In contrast, there are also what are called as water soluble hormones, which could be either small molecules or it could be many polypeptide hormones which are hydrophilic, and because of their inability to cross the lipid membrane, these water soluble hormones primarily interact with receptors which are present on the cell surface, and then this interaction of the ligands with the cell surface receptors ultimately triggers a series of events, ultimately culminating in the activation or repression of the target genes. So, there are two major kinds of signal transduction pathway as far as hormones are concerned. One group of molecules, they simply diffuse into the cytoplasm through the plasma membrane, interact with specific intracellular receptors and activate the expression of or depress the transcription of various genes. There are molecules which are unable to cross the plasma membrane barrier, and as a result they interact specifically with membrane receptors, and this interaction of the membrane receptor with the specific ligands triggers a series of events, ultimately leading to activation or repression of specific target genes. So, in order to regulate the expression of genes in the nucleus through these two classes of hormones, two major signal transduction pathways were evolved. This is what we just discussed just now. Signal transduction by water soluble hormones through receptors located on the cell surface, and signal transduction by lipophilic hormones through receptors present in the cytoplasm or nucleus. So, one is called the extracellular receptor membrane receptors, another is intracellular receptors, which can be present either in the cytoplasm or the nucleus. Now, so this is a cartoon which basically tells you what I have told you so far. For example, there are certain hydrophobic molecules These can be either small ligands or they could be peptide growth factors. Both these categories, they interact with specific receptors which are present on the membrane, and these receptors are often known as transmembrane proteins because they span both, they communicate with both extracellular side as well as the intracellular side or the cytoplasmic side, and they traverse across the plasma membrane often through characteristic domains. We will talk about it a little bit later. So, using this kind of a transmembrane proteins, the molecules, either small ligands or peptide growth factors or a polypeptide growth factors, communicate with specific receptors present on the cells. Like in one case, these are called as G protein coupled receptors or GPCRs, or it could be something like growth factor receptors which actually serve as receptor tyrosine kinases. We will discuss this in later. These kinds of receptors communicate with the small molecules and then transduce the signal through a series of phosphorylation relay mechanism, ultimately leading to the phosphorylation or modification of specific transcription factor, leading to transcription of specific messenger RNAs and synthesis of specific proteins as shown here. In direct contrast, there are certain molecules, especially molecules like steroid hormones, because of their lipophilic nature, they can readily cross through the membrane barrier and then they interact with specific receptors which can be either present in the cytoplasm or they can be present in the nucleus, and then the receptor comes inside, often they dimerize and then bind to specific sequences of target genes, leading to transcription and synthesis of specific proteins. So, whatever I discussed so far I have shown in the form of a cartoon how hydrophilic ligands interact with specific membrane receptors and how hydrophobic molecules simply diffuse through the plasma membrane, interact with specific intracellular receptors, ultimately both the pathways converge into activation or repression of specific target genes. So, I now made a small cartoon to explain to you how signal transduction by water soluble hormones take place through receptors present on the cell membrane. Let us assume this is the plasma membrane of a cell and you have a specific transmembrane receptor which is capable of communicating both outside the cell as well as to the inside the cell. So, once a water soluble molecule, it could be molecules like epinephrine or it could be something like an epidermal growth factor, it could be insulin, it could be glucagon. We will discuss some of these examples in great detail as we go along. So, either macromolecules like huge molecules like the polypeptide growth factors, or it could be small molecules like epinephrine, norepinephrine, so on and so forth. They can interact with some of the specific cell surface receptors, and as a result of this receptor ligand interaction, invariably it results in a conformational change in the receptor molecule, leading to autophosphorylation of the receptor. That is why many of these receptors are known as they serve as protein kinases, but there are exceptions where phosphorylation is not a major mechanism. So, the ligand receptor interaction ultimately leads to the activation of certain relay molecules and one of the most common relay molecules which are used in these signal transduction pathways are what are called as the GTPases or G proteins. Now, these G proteins are basically enzymes which are capable of hydrolyzing GTP to GDP. So, one of the major signal transduction methods involving the signal transduction by water soluble hormones is when the hormone interacts with a receptor, it results in the activation of a GTPase, leading to hydrolysis of GTP to GDP. And these GTPases are capable of choosing their partners depending upon their association with either GTP or in a GDP state, and these GTPs are usually in active state when they are bound to GDP and they become activated when they bound to GTP. And the GTP bound form, they go and associate or activate specific protein kinases, and I have shown here one of the important downstream cascade that happens through this relay mechanism involving GTPases is the activation of an inactive protein kinase to a active protein kinase, and often the signal transduction pathways have this kind of activation and inactivation of a series of protein kinases A, as I have shown here. Protein kinase 1, which is inactive form, gets activated protein kinase 2, active protein kinase 1, and this active protein kinase 1 in turn phosphorylates another protein kinase 2, which then becomes active, and this again can go and phosphorylate another inactivated protein kinase and then convert it into an active form. And as a result of this series of phosphorylation cascades, ultimately the final protein kinase now goes inside the nucleus, which I have shown in the red here, and activates a specific transcription factor by phosphorylation, leading, and these transcription factors can then go either associated with histone acetyltransferases or they can associate with histone deacetylases and so on and so forth, leading to either activation or repression of transcription. So, this is one of the major mechanisms by which molecules which cannot enter the cell interact with specific membrane receptors, activate transcription in the nucleus through a series of enzymes and protein kinases, leading to phosphorylation, dephosphorylation mechanisms, ultimately resulting in the activation or repression of specific genes inside the nucleus. Now, in the case of those molecules, especially the hydrophobic molecules like steroid hormones and so on and so forth, these molecules, unlike the previous ones, can actually enter the cell because they can just diffuse across the plasma membrane, and once they enter the cell, they interact with specific intracellular receptors. As I mentioned earlier, some of these receptors may be present in cytoplasm, or some of the receptors may be present inside the nucleus. Now, once this ligand interacts with this receptor, usually this ligand receptor interaction results in the conformational change inside the receptor, and as a result of this conformational change, the molecule now can often dimerize. They can either form homodimers or heterodimers. There are examples for both kinds, and these dimers now go inside the nucleus, bind to specific response element of genes, and activate the expression of various genes. So, the two cartoons I have shown so far is the two major signal transduction pathways involving receptors acting at either at the cell membrane level or receptor acting either at the through intracellular receptors. Now, things are not as simple as what I mentioned here. The signal transduction pathways are very complicated. Often numerous signals are required to turn on or turn off the expression of a specific gene, and each of these signals is transmitted to the gene by separate regulatory pathways. Because cells are not responding to just one signaling molecule, cells and tissues in our body are often bombarded with multiple signals. So, often in a given cell, it often is bombarded with multiple signals, and multiple signal transduction pathways are getting activated at any given time in any cell type or any tissue type, and so the ultimate activation or repression of genes often is the sum total of all these activation or repression of multiple signal transduction pathways, and often a single gene may be activated by molecules or signaling molecules coming from multiple signaling pathways. So, multiple signaling molecules and multiple transcription factors often act together, leading to synergic activation or repression of transcription of specific genes. So, we will discuss now specific example as we go along to understand how complicated the entire gene expression regulation is, especially when you now come out of the nucleus and start asking the question how events taking place outside the nucleus, outside the cell, influence gene expression programs inside the cell. One of the important things that happens in the signal transduction pathways is the generation of second messengers. The ligand or the hormone which interacted with the receptor and stayed outside is the primary messenger, and often the interaction of this hormone with the ligand results in the generation of what are called as second messengers, and these second messengers ultimately take the signal further down. One of the important significance or advantage of the second messenger is that just when one hormone molecule interacts with the receptor, this single molecule interaction ultimately results in the generation of hundreds or even thousands of second messenger molecules. So, the signal gets tremendously amplified, and that is how even very low doses of a hormone is able to influence gene expression in a big way. Many of these hormones need not be synthesized in huge amounts. Even very small amounts of hormones is able to exert their physiological responses because they can ultimately act through these second messengers. So, interactions happening at the cell membrane, even at a single molecule level, even a single molecule interacting with a single cell surface receptor can generate hundreds or even thousands of second messenger molecule, and thus the signal gets amplified, leading ultimately to the activation of a huge amount of target genes. The magnitude of activation of target gene expression is enhanced. For example, interaction of a single hormone molecule with a membrane receptor can lead to the activation of an enzyme that produces hundreds of molecules of a second messenger. We will discuss in detail how exactly this takes place. But remember, the production of second messengers is a very important adaptation that eukaryotics have evolved, whereby the original signal gets dramatically amplified because all that a hormone has to do is to activate an enzyme. So, that this enzyme can now produce a huge number of these second messenger molecules, and this amplified signal ultimately results in a dramatic increase or decrease in the gene expression. Some of the common second messengers are listed here, like you have what is called the cyclic AMP, you have cyclic GMP, you have diacylglycerol, you have inositol triphosphate, nitric oxide, calcium. These are all the various second messengers that are generated in the cell by various hormone receptor interactions, and each one of them ultimately go and activate specific set of genes, which ultimately results in the synthesis of specific proteins or other macromolecules, and thus gets translated into a specific physiological response. The second messengers that are generated in response to a hormone receptor interaction in turn bind to specific regulatory proteins, inducing a conformational change that ultimately leads to their activation. And once activated, these proteins go on to regulate the activity of a numerous other proteins, including transcription factors inside the cell. So this is one of the basic mechanisms that I would like to highlight, how molecules either interacting with specific cell surface receptors can lead to the generation of specific second messenger molecules, and even one molecule interacting with one receptor molecule can activate, as a result of this phosphorylation cascade, an enzyme which can generate a very high number of second messengers, and thus the original signal gets dramatically amplified. So let us now spend some time to understand how cell surface receptors activate gene expression. We are going to begin with what are called as G proteins, and then we will get into other kinds of cell surface receptors and ask the question what are the various kinds of cell surface receptors which are present in the signal transduction pathways, and what kind of molecules are involved in the activation of these signaling molecules or signaling pathways. Now, water soluble mammalian hormones bind to cell surface receptors that interact with signal transducing heterotrimeric GTP binding proteins or G proteins, which are nothing but GTPases that undergo a conformational change on GTP binding and activate an effector enzyme which generates an intracellular messenger called cyclic AMP. So I am going to begin with one of the major signal transduction pathways that operate in the eukaryotes, and this pathway primarily involves the binding of a hormone, which is basically a water soluble hormone, so that it cannot enter the cell. So interaction of a water soluble hormone with its receptor results in the activation of a GTPase, and this GTPase usually exists in the form of a trimeric protein, and as long as this protein is present as a trimer, it is inactive, and once it is activated by the hormone bound receptor, then the GTPase dissociates, and one of the subunits now become active, that in turn now goes and activates an enzyme called adenylate cyclase, and this adenylate cyclase now starts producing cyclic AMP, and cyclic AMP now acts as a second messenger and initiates a phosphorylation cascade, ultimately results in the activation or repression of specific target genes, or it can also result in the activation or inactivation of specific enzymes in a particular metabolic pathway. So what we are now going to focus in this lecture is about a class of receptors called as trimeric G proteins, which basically contain 7 transmembrane alpha helices. So today's class we are going to now discuss about one particular signal transduction pathway, which ultimately involves the production of cyclic AMP, and we are going to study how cyclic AMP is produced when a specific hormone molecule interacts with a specific cell surface receptor, and what does the cyclic AMP do later. This kind of a G protein coupled receptors or the GPCR signaling is a very robust mechanism of signal transduction pathways that operate in the living cells, especially in higher eukaryotes, and a number of small molecules as well as big molecules like polypeptide hormones make use of these GPCR signaling pathways to activate a repression of enzymes or even transcription factors. Some of the molecules which activate gene expression programs through this GPCR signaling are listed here. These include certain amines, nucleotides, eicosanoids, certain lipid molecules or lipid derivatives, peptide hormones, proteases like thrombin, many glycoprotein hormones like the LH, follicle stimulating hormone, human chorionic gonadotropin, thyroid stimulating hormone, all these big molecules, as well as calcium, glutamine, gamma aminobutyric acid, all these molecules activate gene expression programs or activate enzymes or inactivate enzymes using this GPR signaling pathway. And I have given a nice review which gives a very good overview about this, the mechanism by which all these molecules activate through the GPCR signaling. Now the heterotrimeric G protein, which is a GTPase, is composed of 3 subunits called alpha, beta and gamma. The alpha subunit is about 40 kilodalton in size, and it basically contains a GTPase activity. The beta subunit is about 37 kilodalton in size, whereas the gamma subunit is about 8.4 kilodalton in size, and the beta and gamma subunit together form a dimer. And is a very strong interaction. The beta and gamma subunits interact very strongly with each other, and the only way you can actually dissociate with these two molecules is by actually denaturing the protein. So it is a very strong interaction. So the G protein, which is the key player in the entire GPCR signaling pathway, is a trimeric molecule composed of 3 subunits, alpha, beta and gamma, of which the beta and gamma associate with each other very tightly, and is the alpha which performs a number of regulatory functions. This is a cartoon I have drawn here to actually show you how a typical G protein or a GTPase looks, a trimeric G protein looks. Trimeric G protein is activated by certain receptors which interact with specific water soluble compounds. The activation of these G proteins is primarily carried out by the a receptor present on the cell membrane, and these receptors which activate these trimeric G proteins contain very characteristically what is called as the 7 spanning membrane domains. You can see here the ones which are shown in red here, 1, 2, 3, 4, 5, 6, 7. There are 7 transmembrane helices, and therefore they are often known as 7 spanning receptors. And these receptors play a very important role in translating the extracellular ligand signaling molecule into the activation of a GTPase. We will discuss in detail how exactly this activation takes place. So what are the key domains of a 7 spanning receptor, which is a key player in the G protein signaling pathway? These receptors are basically transmembrane proteins, and as you can show here, there is a transmembrane domain which spans the plasma membrane, and there is an extracellular domain, and there is an intracellular domain which is facing the cytoplasm. And it is through this extracellular domain that the transmembrane receptor communicates with the hormone ligand. It could be either insulin, it could be glucagon, it could be epinephrine. All these molecules interact with this extracellular domain of this transmembrane receptor, and in the intracellular domain you have what is called as a domain that specifically interacts with the G protein of the trimeric GTPase, resulting in its activation. So these are the 2 major key domains of the receptor molecules that communicate with these hydrophilic ligands. So often in the transmembrane domain, the loops which face the extracellular region are often known as the exoloops, and the loops which face the cytoplasmic domain are often referred to as the cytoloops. So remember, these 7 transmembrane domains or the 7 spanning receptors play a very important role in transducing signals by certain hydrophilic molecules which cannot enter the cell through the plasma membrane. And these contain 2 major domains. One domain is the extracellular domain, which is involved in the communication with the growth factors or the small molecules like epinephrine, etc. And you also have a cytoplasmic domain which is involved in the activation of the G protein. The heterotrimeric G proteins are distinct from monomeric G proteins. Again, I do not want to confuse you people. What we will discuss in this class today is about these G proteins or GTPases which are trimeric in nature. Now as we go along, we will also now talk about another specific type of receptors which are different from the 7 membrane transmembrane receptor we have discussed so far, and these are known as receptor tyrosine kinases, which basically exist as dimers, and they activate gene expression by certain growth factors like epidermal growth factor, fibroblast growth factor, and so on and so forth. And these receptor tyrosine kinases activate a different kind of a GTPase, which is a monomeric GTPase. And one of the best example for a monomeric GTPase is a RAS protein, and if you have mutations in this RAS, it results in cancer. So there are 2 kinds of G proteins. One is a trimeric G protein, which are primarily activated by receptors which contain a 7 membrane domain, transmembrane domain. There are monomeric GTPases which are primarily activated by receptor tyrosine kinases which interact with specific growth factors. So in today's class we will discuss primarily about the trimeric G proteins and how activation of these trimeric G proteins result in the activation or repression of specific genes. So the heterotrimeric G proteins which we are going to discuss today are distinct from monomeric G proteins like RAS protein. The heterotrimeric G proteins cycle between an active and inactivative forms, thus acting as molecular switches. So the entire regulation involving G proteins primarily involves the dynamics between the alpha, beta and gamma subunits. the beta and gamma subunits form a very tight complex. As I told you, it is so tight, the only the way you can actually dissociate these two is by actually denaturation. So the beta and gamma subunits form a very tight complex that anchors the trimeric G protein to the membrane on the cytoplasmic side. There is a very nice review in annual review of pharmacology and toxicology about the structure of GPCR. Again, due to problems like copyright and so on and so forth, I could not bring those crystal structure pictures here and then actually show you how understanding the crystal structure actually helped in understanding the various protein-protein interactions involving the GPCR receptor. I suggest you read this review article to actually get a very good idea about the how the structure of the GPCR receptor looks and how it is helps in the cell-cell communication. Now let us now try to understand how the regulation of gene expression takes place involving the G proteins. The G protein becomes activated upon binding to the ligand activated 7 spanning receptor. So once the ligand, the hydrophilic ligand binds to the 7 spanning membrane receptor, the 7 spanning membrane receptor now undergoes a conformational change and it now activates the associated GTP, and it now which is the G protein, which is actually inactive form, exists as a trimer with alpha, beta and gamma subunits together, and in the inactive form the alpha subunit is actually bounds to GDP. So remember, when the G protein is inactive, the alpha, beta, gamma exists as a trimer and the alpha subunit remains associated with GDP. Binding of the receptor liganding complex induces the alpha subunit to exchange GDP to GTP. So once the ligand binds to a 7 membrane transmembrane receptor, it induces it now activates the G protein, and as a result the G protein, instead of now associating with GDP, now associated with GTP. This exchange of GDP to GTP causes the alpha subunit to dissociate from the beta and gamma subunit, allowing the alpha subunit to associate with an effector enzyme, and these effector enzymes usually enzymes like adenylate cyclase, and the GTPase activity of alpha, once the GTP bound alpha subunit associates adenylate cyclase, the GTP is hydrolyzed to GDP and PI, and this energy is now utilized by adenylate cyclase to convert ATP into cyclic AMP. And again the alpha subunit now becomes associated with GDP, and therefore it becomes inactive, and therefore it goes and associates with alpha, beta and gamma unit, therefore signal transduction stops. So the GTP is hydrolyzed to GDP, thereby inactivating the alpha subunit, which in turn inactivates the adenylate cyclase. The alpha subunit bound to GDP reassociates the beta and gamma subunits, and it can again be reactivated by another molecule of ligand binding receptor, and again the entire cycle continues. So you can see the mechanism is very simple. I will explain this whole thing with the cartoon here. This is what is shown here. Here is a hydrophilic ligand, which could be either molecules like insulin receptor, or it could be small molecules like epinephrine, and here is the trans 7 membrane receptor which is present in the plasma membrane, and you have the GTPase which I have shown here, the alpha, beta and gamma subunits in 3 different colors, and the inactive form of the G protein is bound to the GDP here, and you have adenylate cyclase somewhere nearby. This here there is no signal transduction, but let us see the moment now this extracellular molecule binds to its 7 membrane receptor, the alpha, beta and gamma subunit of the G protein, the 7 membrane receptor undergoes a conformational change, and as a result of it the trimeric GTP dissociates, so that only the gamma and beta subunit now associates with the receptor, whereas the alpha subunit dissociates from the trimer, and in the GTP bound form it now activates adenylate cyclase, associated adenylate cyclase and activates, and therefore the energy released by the GTP hydrolysis is now used for converting for the production of cyclic AMP from the ATP by adenylate cyclase. And once this is done, again once the GDP hydrolysis is over, now the GDP bound alpha subunit again becomes inactive, and it goes back and reassociates with the gamma and beta subunit, and adenylate cyclase now becomes inactive. So this is how the binding of a hormone to the cell surface receptor results in the synthesis of cyclic AMP involving a trimeric G protein. So following GTP hydrolysis, the GDP bound alpha subunit of G protein reassociates with the heterotrimeric G protein and is ready to be reactivated by a second hormonal stimulus. So I hope this cartoon has now explained the entire event that takes place. Once a signaling molecules bind to a 7 membrane receptor present on the cell surface, the receptor undergoes a conformational change. As a result, the trimeric G protein now the only the beta and GABA subunit remain associated with the receptor, the alpha subunit dissociates, the alpha subunit now goes and interacts with adenylate cyclase, GTP is hydrolyzed, cyclic AMP is synthesized, and again the GTP bound form of the alpha subunit now again becomes inactive, comes back, reassociates with the beta and gamma subunit, and adenylate cyclase is inactivated. So a burst of cyclic AMP is synthesized. The moment a ligand binds to receptor, the entire event takes place, and one molecule of hormone interacting with the receptor results in the synthesis of 107,000 molecules of synthesis of cyclic AMP during this very brief period. It is given at least 2 articles which discuss in great detail about this entire mechanism of cyclic AMP synthesis involving hormone receptor interactions. So, activation of adenylate cyclase by heterotrymic G proteins increases the concentration of cyclic AMP in the cell, which otherwise is maintained at a very low level by cyclic AMP phosphodiesterase, which hydrolyzes cyclic AMP to 5' cyclic AMP. So, you can see these are all very important regulatory molecules, and so all these things have a very transient half life. They are made and they are immediately degraded. So, when there is no hormonal stimulus, the cyclic AMP levels are actually very low inside the cell, because as soon as the cyclic AMP is made, it is immediately cleaved by enzymes like phosphodiesterase, cyclic AMP phosphodiesterase, which immediately cleaves the cyclic AMP. So, these second messengers, because of their very powerful regulatory activity, their half life is very short and is very highly governed by the degrading enzymes. So, the cyclic AMP thus generated by the activation of adenylate cyclase. Now, the cyclic AMP goes and activates protein kinase A. In the unstimulated cells, protein kinase A exists as an inactive form because of the presence of a pair of regulatory subunits. So, we have what are called as R2C2. Again, we discussed this structure of protein kinase A and regulation when we discuss about in the previous classes, when we discuss the mechanism by which the cyclic AMP response element binding protein activates gene expression. There we actually shown using a cartoon how the protein kinase A exists in an inactive form. You have R2C2. As soon as the regulatory subunit and catalytic subunits are together, it is inactive form, but moment the cyclic AMP levels inside the cell goes up, the cyclic AMP now goes and binds the regulatory subunit of the protein kinase A, and as a result the regulatory subunit dissociates and the catalytic subunit now is free. Now it becomes active. Now it goes and either phosphorylates certain specific enzymes involved in a metabolic pathway, or it can go and go into the nucleus and phosphorylate transcription factors such as the cyclic AMP response element binding protein, or CREB. So, in unstimulated cells, the pKa is in inactive state because of the presence of a pair of inhibitor subunit or regulatory subunits. On cyclic AMP binding to these regulatory and inhibitory subunits, they dissociate from the catalytic subunit, thereby activating the catalytic subunit. The activated catalytic subunits now go and phosphorylate specific serine residues or threonine residues of enzymes such as glycogen phosphorylase kinase, or they can go and phosphorylate transcription factors such as CREB. Now, what is the consequence of this? As a consequence of this, when there is an increase in the cyclic AMP levels inside the cells, and when there is an activation of protein kinase A inside the cells, it can either result in the activation of metabolic enzymes, or it can result in the activation of transcription factor. That is what is shown here. The pKa on phosphorylation, when phosphorylated by pKa, the glycogen phosphorylase kinase phosphorylates or activate another enzyme called glycogen phosphorylase, and this enzyme now involved in the breakdown of glycogen in muscle cells to glucose 1 phosphate. For example, this is what happens during glucose metabolism. When glucagon is produced, the glucagon binds to this corresponding glucagon receptor, which is a 7-membrane receptor, and this results in increase in cyclic AMP. Cyclic AMP now comes and activates protein kinase A. Protein kinase A now goes and phosphorylates the glycogen phosphorylase kinase. Now it becomes active. Glycogen phosphorylase kinase now goes and activates glycogen phosphorylase. That goes and now breaks down. That is now involved in the breakdown of glycogen, leading to the production of glucose 1 phosphate in muscles. So, this is how the body responds whenever there is elevated levels of glucagon. So, when there is no glucose in the cells, in the circulation, in the blood, the glucagon is released by pancreas, and the glucagon now comes and binds to the 7-membrane receptor, and that now leads to the synthesis of cyclic AMP, leading to the activation of glycogen phosphorylase, ultimately increase in the blood concentration of glucose. Now, the same protein kinase A, in addition to activating the enzymes involved in the glucose metabolism or glycogen metabolism, can also now go inside the nucleus and phosphorylate the cyclic AMP responsible and binding protein, which now goes and can interact with the histone acetylase I had histone acetyl transferase, especially this Krebs binding protein or the CBP, also known as the P300, and as a result there is a faster recruitment of transcription factor leading to activation of gene expression. So, you can see how interaction of these hydrophilic or the lipid insoluble molecules like glucagon or epinephrine or even insulin and so on and so forth molecules by interaction with specific membrane receptors can result in the activation of a GTPase, result in the synthesis of cyclic AMP. Cyclic AMP now activates protein kinase A, and this protein kinase A either can activate metabolic enzymes or it can activate transcription factor leading to activation or repression of genes. Now, this kind of a G protein signaling is used by a number of used in number of physiological responses. I have listed some of these things here. Smell and taste involves G protein signaling. There are more than 1000 types of this kind of a 7 membrane transmembrane receptors which are involved in sensing our smell and taste. The way we perceive smell, the way we taste things, all these things involve G protein signaling. Perception of light involves these 7 membrane receptors. Rhodopsin, one of the major receptors involved in perception light, is a 7 membrane transmembrane protein. Neurotransmission, function of exocrine and endocrine glands, chemotaxis, endocytosis, control of blood pressure by epinephrine, non-epinephrine, and so on and so forth. Embryogenesis, development, cell growth and differentiation, HIV infection, oncogenesis. You can see a number of physiological processes make use of this kind of a G protein signaling involving a trimeric G protein receptors, ultimately leading to the intracellular increase in second messengers like cyclic AMP, leading to activation of specific G protein signaling pathways. And a number of nice reviews I have listed here which you can go through to understand how G protein signaling plays a very important role in the activation of a number of in the development number of physiological responses. Mutations in the GPRs, the G protein coupled receptors, result in constitutive signaling. If you have mutations in the G protein subunits, then the alpha subunit becomes constitutively active, and as a result there is no regulation. The GTPase is constitutively active, therefore it constitutively interacts with the adenylyl cyclase. The cells continuously producing cyclic AMP, and as a result there is a continuous activation of the enzymes of metabolic pathways or continuous activation of transcription factor, and it can have disastrous consequences. And when these kinds of things happen, it results in a number of diseases, some of which I have mentioned here, like familial hypothyroidism, familial male precocious puberty, Janssen metaphyseal chondroplasia, congenital night blindness, hyperfunctional thyroid nodules, familial non-autoimmune hyperthyroidism, and so on and so forth. There are nice reviews I have listed here which discuss how defects in G protein signaling can lead to a number of diseases. I have also given a table here which gives a much more detail about how defects in some of the G protein coupled receptors can lead to a number of diseases, what kind of point mutations have been shown to be involved in these G protein coupled receptors, and what kind of diseases it manifests in. There is continuous like adrenergic receptors, dopamine receptors, all these are G protein coupled receptors, and when you have mutations in these things, it results in the manifestation of a it manifests in the form of a specific disease. So, what we discussed today is about one major signal transduction pathway which involves interaction of certain water soluble molecules with G protein coupled receptors or GPCRs, which are 7 membrane which contain a 7 membrane transmembrane domain, and this interaction activates a GTPase, which is a trimeric protein, and the trimeric protein result, this activation involves dissociation of the alpha subunit from the beta gamma subunits. This alpha subunit now goes and activates adenylyl cyclase, that results in the synthesis of cyclic AMP. Cyclic AMP now goes and activates protein kinase A, that ultimately now either activates enzymes of metabolic pathways or activates genes of a particular transcription pathway. There are many other signal transduction pathways which involve smaller G proteins such as RAS. There are intracellular receptors, there are serine protein kinases which are involved in signal transduction pathways, there are receptor tyrosine kinases, and there are phosphatases which negate the action of the kinases. There is calcium signaling, nitric oxide signaling. What we will do in the next few classes is to take all or at least some of these as examples to understand how activation of each one of the signal transduction pathways ultimately results in the activation or repression of specific gene expression programs. I think I will stop here. So, what I have discussed in this class today is to introduce you to the area of signal transduction, and the message I have conveyed to give you is that activation or repression of transcription of genes inside the nucleus is not just confined to nucleus alone. This whole thing happens in response to a variety of stimuli that the cell encounters during its life. So, depending upon whether the signaling molecules are capable of entering the cell and interacting with specific intracellular receptors, or whether they interact with specific membrane receptors, both these mechanisms ultimately results in the generation of either second messenger molecules, or it involves in the activation of specific phosphorylation cascades, and as a result specific transcription factors are activated, ultimately resulting in activation or repression of specific genes inside the nucleus. And one signal transduction pathway which we discussed in detail today involves the G protein coupled receptors, through which trimeric G proteins are activated, and this trimeric G protein in turn activates enzyme like adenylyl cyclase, resulting in synthesis of cyclic AMP, which then phosphorylate, which then activates protein kinase A, which in turn activates specific gene expression programs. So, in the coming classes we will now take up a few more examples of the signal transduction pathways and understand how molecules interacting either with cell surface receptors or with intracellular receptors ultimately activate or repress transcription of specific genes. Thank you.